Welcome to our Bonito Lab Training video on confocal laser scanning microscopy of Arabidopsis thaliana roots with the Olympus FluoView FV10i microscope. I'm Julian Lieber. Retrieve your plates from a growth chamber and open them up with lid side down to drain any condensed water. Cut the shoots from the roots using a sharp, sterilized scalpel just below the leaf rosette. Pull the roots from the plate using sterilized forceps. Place one plant's roots in a negative control tube to which you will not add stain. Take additional roots, this time needing to scrape away extra agar, and place into a new tube. We will add phosphate buffered saline to the negative control tube. Then we'll add stains dissolved in the same buffer to the treatment sample. The only difference in treatment is the addition of stain to the sample. Here we are using 25 micrograms per milliliter WGA 640R and 1 milligram per milliliter calcifluor white. Open the tubes, cover them in foil, and place in the vacuum chamber. Turn off the lights to reduce breakdown of the stain. Attach the vacuum pump and apply vacuum for 30 seconds before releasing. Repeat twice more. Remove the tubes and close them, then cover in foil. And place on a shaker for 30 minutes at about 60 RPM. Turn on the equipment in the order shown. There's a switch on the back of the microscope's power supply. The microscope is ready to use when the flashing green light becomes solid. Clean your glass slides with 70% ethanol and a lab wipe. First, we are going to process the negative control sample without stain. It is not light sensitive, so we can have the lights on for this step. Place the roots on a slide with clean forceps. Add a little water and spread the roots out into a single layer if possible. Slowly place a cover slip on the slide. Retrieve the sample holder from the microscope body. Place the inverted slide, cover slip down, in the holder. Use the holder clips to secure the slide and position the center of your sample to be aligned with one of the numbered ticks. Place your cover slip at a slight angle to make it more secure. Place the sample holder into the microscope. Open the software and enter your username and password. You will need to request a profile before use. From the dyes listed, select the ones that match the excitation and emission wavelengths of the stains in your sample and press apply. Press Start, select Use Fluorescence if desired, and press OK. Register the container corresponding to the tick mark on the sample holder, then select Automatic Registered Container and hit Start. The microscope will first autofocus, then scan the sample at low resolution. Hit Next after this is complete. Find a location of interest on the scan map and click on it to place the red crosshairs there. You can zoom in if needed. Go to Other Parameter and select 512 by 512 and high speed times 1. Press Repeat and a preview image will be acquired. Each top panel displays one stain, in this case calcifluor white in teal and WGA in blue. The gray one is phase contrast and the bottom right is a merged image. Click on one panel, then reduce the sensitivity until the image appears dark. Repeat for the other fluorescent panels. Because this is the control sample, we should have a baseline fluorescence without the stains added. Record the sensitivity values for later. We can then change the focal depth to ensure that no signal is present. Once you are satisfied, go back to Acquire Map Image and prep the stain samples. Manipulate the samples with stain in the dark. You will need to again place the roots on a slide, add water, spread them out, and add a cover slip. Change the slide in the sample holder, again noting the tick mark number. Replace it in the microscope. Press Start, allow the microscope to re-autofocus, and press Next when complete. Find a location on the sample map, then press Repeat. Set your sensitivity values to what you recorded on the control sample. Navigate around the sample image by double-clicking, which centers the image, or by clicking and dragging. You can change the zoom level with the slider. You can also remove or add channels to the merged image by selecting or deselecting the tabs above the panels. Change the focus position with the left slider under Depth. To move to a higher objective, press Repeat, then press the 10x button. Once the lens moves into position, you will need to add a drop of Olympus immersion oil on the port labeled Oil. Press OK, and the objective will move into place. Now we are viewing with the 60x objective. You will also need to set the sensitivity values again. As we move the focus up and down, we can see that the 60x slices are much thinner due to its smaller aperture. Each focus depth shows a thin cross-section of the sample. In order to capture a 3D series of images, called a Z-stack, set the start and end depth values just above and below the focus depth of the features you are interested in. Change the image size to 1024 by 1024 and the speed to a higher value depending upon how long you want your stack to take and the quality that you want. Name your photo descriptively and start the series. The 1 repeat time tells you how long the series will take to acquire. After you've acquired an image, click on the Review tab on the upper left of the screen. Refresh your directory and click on your recent images. You can change which channels are shown with the left tabs, and use the slider to view each slice of a Z-stack. Click on the Color tab in the upper left, then you can change the color intensity and noise by setting the maximum and minimum values. You can also change the colors to make the features easier to distinguish. We are first going to export the image as a multi-TIFF, which allows for other uses such as 3D reconstruction in 3D Slicer or maximum intensity projection in Image J. We can also capture and export a single slice, here with the scale bar added, then export just that slice as a JPEG. JPEGs should not be used for later processing because they lose information relative to a TIFF. Before finishing, clean the oil off the 60x objective with lens paper and switch back to the 10x objective. Save or dispose of your sample, then either clean your slide or place it in the sharps waste. Cover slips should go in the sharps waste. Exit the software and replace the sample holder. Turn off the devices in the order that you turn them on. Thank you for watching.